Cool. Back to, I think it was Noem's question. The origin of species is a reference to a book by Charles Darwin, and we'll talk about that later. Okay, what we're going to be covering today: heredity, evolution, natural selection, speciation, taxonomy, and then prokaryotes. The reason we're covering heredity is because we took note of your feedback form, and the few people who said that they wanted to learn more biology, we had one person who said they wanted to learn more about heredity. So we do take your feedback into consideration. If you have not filled out the feedback form, please fill it in. So some just key terms about heredity. Genes are fragments of DNA, and then alleles are going to be the specific form of a gene. This should be reviewed, by the way, for most people. And then the genotype is the set of genes an organism carries. For example, you could have a trait for blonde hair or a trait for brown hair. So genotypes are generally all traits. But then a phenotype is what you actually see and how that trait is or how that gene is expressed. So having a trait for brown hair does not necessarily mean you will have brown hair. But in your phenotype, if you can like have brown hair, and you literally have brown hair. Used to determine most hereditary genes, because the Punnett squares are very simple ways to determine how you are going to inherit your genes. And quick disclaimer, Punnett squares only work for Mendelian traits, which means that these traits are not like linked together, or which means that they won't be crossed over together when you go through meiosis. This is really important because some traits, they are linked, and so Punnett squares don't work for them. And so some of you probably have seen a Punnett square before, and you label the dominant and recessive traits, which are both alleles. You label the dominant trait with a capital letter and recessive traits with a lowercase letter. So homozygous means that the two traits have the same allele. So for example, RR, if you were labeling, if the trait was called R, I guess. So capital R, or lowercase r and lowercase r will be homozygous, while capital R and lowercase r will be heterozygous. So really quickly, as a quick review, for those of you who do know Punnett squares, try crossing a heterozygous genotype with a homozygous genotype that's recessive. And yeah, we'll take like 10 seconds for that. Cool. Okay, this is what the Punnett square should look like. Of course, you can flip the top of the rows in the column. So for example, you could have lowercase r and lowercase r here versus uppercase r and then lowercase r here. So that works. But in the end, you're always going to get the same 4 combinations. So here you would see that the r and the r becomes rr, and it's because a dominant is always going to go over a recessive. The trait that's going to show up is the capital R. Same for this one. Here you have 2 recessive traits, so then the recessive trait will show. And then same for this. So here you see 50% heterozygous and 50% homozygous recessive. Questions about how a Punnett square works? No? Cool. Okay. So to evolution and natural selection. So there have been a couple theories of evolution throughout history. First, we have Aristotle, who believed that life forms could be organized in a ladder, and basically he thought that evolution never happened. So he would see that there are organisms that go in increasing complexity. For example, you have bacteria, although they didn't know about bacteria back then. But if he knew about bacteria, he would have thought that bacteria are really simple life forms. And then he would go up to like humans, which are made out of like millions of cells, trillions, billions, yeah, and are very complex life forms, you could say. But then later on in 1809, Jean-Baptiste de Lamarck, we usually call him Lamarck, he published the hypothesis that, for example, if you used a body part a lot, such as your neck, and you stretched it a lot, over the course of your life, your neck would grow in length. and strength, and then this like long neck characteristic will be passed on to future generations. So basically Lamarck thought that, for example, giraffes used to have shorter necks, but then one giraffe in particular decided to stretch its neck a lot, and then slowly its neck became longer, and then after it had offspring, its offspring inherited that long neck. But then after that, Darwin came in and discovered that that was not true. So in 1859, he published his book on the origin of species by means of natural selection, which generally is abbreviated into the origin of species, which is by chance or purposely the title of this presentation. And Charles Darwin published his idea of natural selection. So essentially what natural selection is an idea of descent. Sure.
Could you go back for a second, please?
Yeah. Tell me when you're ready to move on.
I'm ready, thanks.
Cool. Natural selection. What natural selection is, it's descent with modification. So this means that over generations, you accumulate diversity. For example, because of radiation or other just normal mutations in your genes that are caused by accidental like crossing overs during meiosis or something like that, you're going to accumulate differences in in your genome or like a population's genome. And what happens is that if that genome is good for surviving in the habitat, or it increases your chance of survival, or it increases your reproduction rate, then that gene is going to be passed on, right? Because you have more offspring to pass on that gene. So this idea is based on the fact that all species have overproduction. So you reproduce more offspring than survive to adulthood, because unfortunately there's like disease and famine and bad stuff happen, and not everyone's going to survive to adulthood. So then in this process, the species that have less good traits to survive in a habitat, that's probably not proper grammar. Anyway, that have less good traits to survive in a habitat are going to die off with a higher percentage, right? Because, for example, if you were living in water and you didn't have gills, that would be rather unfortunate, and there's a very high chance that you would die. So what happens is that those animals, organisms that do have gills, they're going to survive. They're going to pass on the gene for gills to their offspring. And this is what's essentially called survival of the fittest, which is something that probably most of you heard. And fittest, but what it means, it's not necessarily like the most strong or the tallest. What is survival of the fittest means is the fittest means the most suitable to the habitat that the organism is living in. So some examples of evolution we have seen so far are drug-resistant bacteria. For example, MRSA, or methicillin-resistant Staphylococcus aureus. So Staphylococcus aureus by itself is not resistant to methicillin, but unfortunately, because we used a lot of methicillin and antibiotics like these, one bacteria mutated, came up with a mutation that made it methicillin resistant. And then when methicillin killed all of those bacteria that were not resistant, the one that was resistant was left, it reproduced, and now we have MRSA, which is rather unfortunate because it's a very deadly bacteria. And also, example of evolution would be homologous structures. So they are anatomical resemblances between closely related species. So for example, all mammalian forelimbs have some resemblance and have the same structure and bones. For example, we have the same structure and bones as whales, actually. And additionally, for example, vestigial structures, which are structures that have no use but are still present in animals. For example, humans have a tailbone, although when you grow up, you don't have a tail, hopefully. But when you're an embryo, human embryos do have tails, and that's a vestigial structure that we see. Another example of evolution is going to be analogous structures. So those are anatomical resemblances, but they evolved independently from each other. And the reason that they evolved independently is because, for example, there are similar habitats across continents, but these continents are like separated by ocean in the case of the sugar glider versus the flying squirrel. For example, the flying squirrel lives in North America, sugar glider lives in Australia. They're separated by ocean. And so these two animals, they evolved independently, but because they lived in similar habitats, it was very beneficial for both of them to be able to glide through trees and have that flap of skin between their forelimbs and their hind limbs. And the last example of evolution is endemic species, the species that can only exist on a certain island or land area, although the island is going to be near other land. And the reason for this is because that island is going to have its own special habitat. So although that island is near other land with different habitats, we're going to see species that only survive on the island because they're only adapted to that island's specific habitat. Can I move on? I will take that as a yes. So you see in the above picture, these are the examples of homologous structures in all mammals. So we see that the bone structure is essentially the same. The bone structure, each color represents one bone. So you can see that we all have humerus, radii, ulna, carpals, metacarpals, and phalanges, even though you might not think that humans are very similar to whales or bats. Underneath here, we see analogous structures. So you see marsupials and flying squirrels, and they look remarkably similar, although they evolved 100% independently of each other. And in fact, sugar gliders are marsupials, while flying squirrels are placentals. And essentially what placentals are is that when their offspring is an embryo, it is connected to the mother through a placenta. But you will notice that, for example, marsupials, the embryo eventually comes out into the pouch. It doesn't stay inside the mother for the entire development. Okay, so another important part about why we have so many species today is the idea of speciation. So there are a lot of definitions of what a species exactly is, but some of the main ones are the biological species concept, which means that a species is the group of organisms that can produce fertile offsprings among themselves. And if this group of organisms cannot produce fertile offspring with other groups, then these two groups are separate species. But unfortunately, this only works for species that produce through sexual means. It doesn't work for asexual because asexual reproduction is just copying yourself. And then you have the morphological species concept. So this one, it distinguishes between body shape and body structure. And this works for sexual and asexual species. What's unfortunate about this one is that it relies on really subjective criteria. For example, some people might think that a giraffe's neck is not that long, versus others might think that it is very long. And so this type of subjective criteria can lead to some scientists to determine that these two animals or these two populations are one species, while others may believe that it's a different species. And then you have the ecological species concept, and this one distinguishes through ecological niche. So what a niche is essentially it's what an organism performs in its environment or its habitat. So for example, we have decomposers, or we have predators, or producers, stuff like that, and those are all niches. And then there are a lot of other different definitions, and scientists are always constantly thinking of new definitions for what a species is. There's like 20 more, I think, so far. So we are going to be focusing on the biological species concept because it's the most widely used for sexual species, and it's kind of the most common. And it's the one that scientists have spent the most time kind of defining out. All right, there. So there are going to be to create a new species and to have a new species defined by the biological species concept, you're going to have reproductive barriers, because then the definition of a new species under that concept is that two different groups can't reproduce. So you can have different types of reproductive barriers. For example, you can have pre-zygotic barriers, so they just prevent mating. And for example, some of these can be habitat isolation, so different habitats altogether. Temporal isolation, which means different times for breeding, mating, or behavioral isolation, which means different mating behaviors. And then post-zygotic barriers are barriers that prevent offspring from developing into a viable fertile adult. And these can be, for example, reduced hybrid viability. So the genes are going to interact in ways that cause weird features. You can have reduced hybrid fertility, which means that the offspring do grow up, they are healthy, but they can't have children. And then you can have hybrid breakdown. So your first generation offspring are going to be able to have children, but then your second generation, it just doesn't work out and the offspring are not healthy. And then we have types of speciation. So how exactly can speciation occur? We have allopatric speciation, which basically means that you have a geographic barrier. So for example, a mountain or a canyon or a valley or canyon and valley are kind of the same thing, or a river, some body of water, stuff like that. And so allopatric speciation is special in that, for example, a canyon might be really hard for an ant to cross, but for a bird, a canyon isn't going to be a barrier. So allopatric speciation depends on the ability of the organism to move. Then we have sympatric speciation, which is going to be a lot less common than allopatric speciation. And what sympatric speciation is, it's speciation that occurs in populations that are living in the same geographic area. And the reasons that can be caused by are, for example, polyploidy. So when meiosis occurs, there's an extra set of chromosomes in one of the gametes. And then when it becomes a zygote, that organism is going to have an extra set of chromosomes. And this is really common in plants because generally in animals, once you have polyploidy, that offspring is going to die. But in plants, polyploidy works. So for example, strawberries are like octoploid. Then it can also be like sexual selection. So for example, the birds of paradise, it's like how you mate, and it's kind of the behaviors, mating behaviors that cause a different type of speciation. And then lastly, you can have habitat differentiation. So for example, if suddenly in this habitat there came apples, and then this type of fly that previously only laid eggs or only fed on blackberries or something like that, one fly might decide to go out and start feeding on apples, and that over time might cause a new species. So we have a couple of models of speciation. For example, we have punctuated equilibria. So what this is, or you can hear it called punctuated equilibrium, but essentially what it is, it's a pattern in the fossil record, and what happens is you have a new species that suddenly appears, and then that species stays the same for a really long time, usually millions of years. And then suddenly that species just goes extinct. So you just see these sudden changes in the fossil record. So where punctuated equilibrium occurs, we call that the punctuated model. So it's really quick speciation, like suddenly you have a new species and then suddenly that species is gone. But then we also sometimes see the gradual model. So where species are going to slowly change over time, and this is gradual speciation, and we see both models occur. So sometimes in some cases there are going to be rapid speciation, sometimes in some cases it's going to be gradual. And scientists haven't really figured out why there is going to be a punctuated versus gradual. But for example, sudden extinction events are probably going to cause punctuated equilibrium. Okay, and then taxonomy. This should also be reviewed. So some key terms about taxonomy. So taxonomy is the study of organisms and how they are named and classified. So in taxonomy, we use binomial nomenclature. So that is the format of the scientific name of organisms, and it was developed by Carolus Linnaeus. So what the format is you have the genus and the species. The genus is going to be capitalized, the species is going to be lowercase, and generally the entire scientific name is going to be put in italics. So for example, Canis lupus is wolf, the scientific name for the wolf. And then what a taxon is a taxonomic unit. So for example, in the level of the genus, you're going to see Canis is going to be the taxon at the genus level. Or for example, Homo sapiens, Homo is going to be the taxon at the genus level. Homo sapiens is humans. Hopefully everyone knew that. To diagram taxonomy or to diagram evolution and speciation throughout history, we use phylogenetic trees. So they are diagrams that represent the evolutionary history of an organism. And so you can see here that each branch point is going to represent a common, or each point at the branch is going to represent a common ancestor. And then these two branches are sister taxons. So this is a part of the taxonomic tree, and what this represents is the chordata. And so what it is, it's those that have a notochord, the thyroid gland, and then there are two other characteristics, but you can think of it basically as vertebrates. So except for this branch, which doesn't have an actual vertebrate, but some people do call it a vertebrate. Well, essentially this branch, it has notochords, so it's kind of a predecessor to the vertebrate, you could say. Okay, and then this is your order of the taxonomic classification. Please remember this, be able to remember in order. Usually it's easier to go top down, but this is definitely going to come up in Science Bowl. It comes up pretty frequently, to be honest. So I will stay on here so people can like list it down. Does anyone know, can someone list for me the 3 domains? Can y'all hear me?
Can hear you.
Okay, can someone list for me the 3 domains? Does anyone know the 3 domains?
Can you show me how that?
Bacteria and eukarya.
Wait, can you repeat that? Sorry.
Archaea, bacteria, and eukarya.
Yeah.
Okay.
Y'all should also remember that. Okay, moving on. So we are going to also go over prokaryotes. And this is, prokaryotes cover 2 of the 3 domains that we just mentioned. Prokaryotes are going to cover archaea and bacteria. And prokaryotes are also kind of the beginning of life. They are quite literally the origin of species, because we all start out with, life started out as prokaryotes. And then it became, or eukaryotes evolved, essentially you can think of it as. So one of the domains under prokaryotes are bacteria, or I think in some textbooks, very old textbooks, they're called eubacteria. So that's good to note. And so bacteria have 2 types that you can split them into, gram-positive and gram-negative. So gram-positive bacteria have cell walls with a thick layer of peptidoglycan. And what peptidoglycan is, it's cross-linked sugar polymers. And then gram-negative bacteria have cell walls with less peptidoglycan, but they are more structurally complex because they're going to have an outer membrane. And gram-negative bacteria are really unfortunate because they are resistant to a lot of the antibiotics because the outer membrane impedes the entry of drugs. So gram-negative bacteria, most of the antibiotic-resistant bacteria that you are going to see are going to be gram-negative. And gram-negative bacteria become a pretty big problem in recent years. So because bacteria are prokaryotes, they are going to have a single chromosome, and that is located in the nucleoid. So what the nucleoid is, it's a general area where the chromosome is located. Because they don't have membrane-bound organelles, so they don't have a nucleus. And then bacteria are also going to have plasmids, which are really short rings of independent DNA from the single chromosome. And what's really interesting about plasmids is that some bacteria can transfer plasmids between bacteria. So it's kind of like you can transfer part of your DNA. And so that also leads to rapid mutations and rapid differences in the genome. Also, bacteria are going to reproduce really rapidly, or I guess you can call it reproduction, but it's asexual reproduction, so it just duplicates itself. But because it's so quick, there's also going to be a lot of mistakes when copying the DNA. And so this can also lead to a lot of mutations. And what happens is because these bacteria, they change really, really quickly. That's why, for example, we annually have to change the flu shot or stuff like that, because bacteria are mutating and evolving so rapidly that you constantly have to find new antibiotics or new vaccines. And another way that bacteria are going to get new DNA is they can actually take DNA from their surroundings. And this is called transformation. So the other domain under prokaryotes is archaea, and you can sometimes see it called archaebacteria. I think that is also seen in very old textbooks. So under archaea, you have the extremophiles, and this is really important. Please note that not all archaea are extremophiles, but most extremophiles are part of the archaea domain. Now, a lot of people got this wrong in the Science Bowl tryouts for biology, so please do take note of this. So we have some different names or we can categorize extremophiles. So for example, halophiles are extremophiles that can withstand really high salt percentages. And then thermophiles are ones that can withstand very high heat. And then there's another one called chemophiles, which can withstand a lot of different chemicals that would actually be toxic to other organisms. And then we also have the methanogens. So these are archaea that release methane, and they do this by oxidizing hydrogen into carbon dioxide. And this produces energy and methane. And what's really important about methanogens are they are really strict anaerobes, which means they are poisoned by oxygen, so they cannot live in environments with oxygen. And then under archaea, you're also going to see the TAC supergroup. I am not going to pronounce these because they are very long. And to be honest, in Science Bowl, if you just go by what it looks like, the reader will probably accept it because the reader probably doesn't know how to pronounce it either. But what's important about the TAC supergroup is going to be because most scientists recently have think that the TAC supergroup is the sister group to eukaryotes. So it's what eukaryotes may have evolved from. And that provides a really important link to evolution and to the start of life. So that's the end of this presentation. Does anyone have any questions? No? Okay, then you guys can have like a 10-minute break. If anyone has any questions or any slides they want me to go back to, feel free to ask right now. We will also be posting this presentation in the Google Classroom, so you can also look at that. If you have not filled out the feedback form, please fill it out. If you have not joined the Google Classroom, please tell us, let us know, because you're missing out on a lot of material.